this session, we are going to see the uses of translocations. Changes that affect the structure of the chromosome can cause problems with growth, development and function of the body systems. These changes can affect many genes along the chromosomes and disrupt the proteins made from those genes. Structural changes can occur during the formation of egg or sperm cells, in early fetal development, or in any cell after birth. Pieces of DNA can be rearranged within one chromosome or transferred between two or more chromosomes. The effects of structural changes depend on their size and location and whether any genetic material is gained or lost. Some changes can cause medical problems, while others may have no effect on person's health. Changes in chromosome structure include basically translocations. A translocation occurs when a piece of one chromosome breaks off and attaches to another chromosome. This type of rearrangement is described as balanced if no genetic material is gained or lost in the cell. If there is a gain or loss of genetic material, the translocation is described as unbalanced translocation. The next one is deletions. Deletions occur when a chromosome breaks and some genetic material is lost. Deletions can be large or small or can occur anywhere along a chromosome. Duplications. Duplications occur when part of a chromosome is copied or duplicated too many times. This type of chromosomal change results in extra copies of genetic material from the duplicated segment. The next type of translocation is inversions. An inversion involves the breakage of a chromosome in two places. The resulting piece of DNA is reversed and reinserted into the chromosome. Genetic material may or may not be lost as a result of the chromosome breaks. An inversion that involves the chromosome's constriction point or the centromere is called a pericentric inversion. An inversion that occurs in the long Q arm or short P arm and does not involve the centromere is called a paracentric inversion. Let's see the isochromosomes. An isochromosome is a chromosome with two identical arms. Instead of one long Q arm or one short P arm, an isochrome has two long arms or two short arms. As a result, these abnormal chromosomes have an extra copy of the same genes and are missing copies of the other genes. Let's see what dicentric chromosomes are. In this, unlike the normal chromosomes, which have a single constriction point, the centromere, a dicentric chromosome contains two centromeres. Dicentric chromosome results from the abnormal fusion of two chromosome pieces, each of which include a centromere. These structures are unstable and often involve a loss of some genetic material. Let's see what are ring chromosomes. Ring chromosomes usually occur when a chromosome breaks in two places and the other ends of the chromosome arms fuse together to form a circular structure. The ring may or may not include the chromosome's constriction point, that is the centromere. In many cases, genetic material near the ends of the chromosome is lost. Many cancer cells also have changes in their chromosome structure. These changes are not inherited. They occur in somatic cells, cells other than the egg or the sperm, during the formation or progression of a cancerous tumor. In genetics, a chromosome translocation is a chromosome abnormality caused by the rearrangement of parts between non-homologous chromosomes. A gene fusion may be created when the translocation joins two otherwise separated genes. It is detected on cytogenesis or karyotype of affected cells. Translocations can be balanced in an even exchange of material with no genetic information extra or missing, and ideally fully functionality, or unbalanced where the exchange of chromosome material is unequal, resulting in extra or missing gene. The below figure explains how the chromosome architecture changes before and after translocation. The figure explains the chromosomal reciprocal translocation of the 4th and 20th chromosome. Reciprocal translocations. Reciprocal translocations are usually an exchange of material between non-homologous chromosomes. Estimates of incidence range from about 1 in 500 to 1 in 625 human newborns. Such translocations are usually harmless and may be found through prenatal diagnosis. However, carriers of balanced reciprocal translocations have increased risk of creating gametes with unbalanced chromosome translocations, leading to miscarriages or children with abnormalities. Genetic counseling and genetic testing are often offered to families that may carry a translocation. Most balanced translocations carriers are healthy and do not have any symptoms, but about 6% of them have a range of symptoms that may include autism, intellectual disability, or congenital anomalies. A gene disrupted or dysregulated at the breakpoint of the translocation carrier is likely the cause of these symptoms. It is important to distinguish between the chromosomal translocations occurring in the gametogenesis due to errors in meiosis and translocation that occur in cellular division of somatic cells due to errors in mitosis. The former results in a chromosomal abnormality featured in all the cells of the offspring, as in translocation carriers. Somatic translocation, on the other hand, result in abnormalities featured only in the affected cell line, as in the chronic myelogenous leukemia with Philadelphia chromosome translocation. Reciprocal translocations are nearly always spontaneous, that is, they occur during the formation of the egg or the sperm, or during or shortly after conception. Therefore, neither parent has the chromosome change in some cases. However, one parent may have a balanced reciprocal translocation themselves that causes no problem for themselves, but may cause problems in the pregnancy or with the baby's chromosomes makeup. Balanced reciprocal translocations. In this type, the individual with a balanced reciprocal translocation will usually have the correct amount of genetic information for normal development, simply rearranged differently from the usual. They are a translocation carrier when a parent is an unaffected carrier of a balanced translocation. Their child may have the same balanced chromosomal translocation in their cells as their parent and is also unlikely to be affected by the chromosome rearrangement. Even though the parents themselves may not be affected, there is an increased chance that there will be reproductive consequences due to the child receiving the unbalanced chromosome complement, that is, the child has more or less chromosomal material than usual. For example, one of the parent is a couple carries a balanced translocation between the chromosomes 1 and 9 as shown below. This unbalanced reciprocal translocation had no effect on the parent who is the translocation carrier, as well as the genetic information was present. A person with this chromosome arrangement has a balanced translocation with the equivalent of 2 chromosomes, each of the numbers 1 and 9. The child, however, received a normal copy of chromosome 9 from each parent, one normal copy of a chromosome 1 and the translocated chromosome 1 with an additional chromosome 9 material and unbalanced translocation. a diagrammatic representation of the unbalanced autosomal translocation between the chromosome 1 and the chromosome 9 in a child where one parent has the balanced translocation. Impact of the unbalanced translocation in the child. The result is that the child has 3 copies of the material from the chromosome 9 depending on the information contained in the additional chromosome 9 material. The child may have a range of problems or may not be affected at all. The nature of these consequences depends on the particular chromosomes involved and the size of the translocated material. It is not possible to outline all of this in this fact sheet. It is, however, important for balanced translocation carriers and their partners to seek specialist genetic advice prior to conception. Let's see what's Robertsonian translocations. In Robertsonian translocation is a type of translocation caused by breaks at or near the centromeres of the 2 acrocentric chromosomes. The reciprocal exchange of parts gives rise to one large metacentric chromosome and one extremely small chromosome that may be lost from the organism with little effect because it contains so few genes. The resulting karyotype in humans leaves only 45 chromosomes, since 2 chromosomes have fused together. This has no direct effect on the phenotype, since the only genes on the short arms of the acrocentric are common to all of them and are present in variable copy number, which is also said as nuclear organizer genes. Robertsonian translocations have been seen involving all combinations of acrocentric chromosomes. The most common translocation in humans involve chromosomes 13 and 14 and is seen in about 0.97 for 1000 newborns. Carriers of Robertsonian translocations are not associated with any phenotypic abnormalities, but there is a risk of unbalanced gametes that lead to miscarriages or abnormal offspring. For example, carriers of Robertsonian translocations involving chromosome 21 have a higher chance of having a child with Down syndrome. This is known as translocation Downs. This is due to the missegregation or the non-disjunction during the gametogenesis. The mother has a higher 10% risk of the transmission than the father with 1%. Robertsonian translocations involving chromosome 14 also carry a slight risk of uniparental disomy 14 due to trisomy rescue. Balanced Robertsonian translocations. Balanced Robertsonian translocations also generally do not result in physical or developmental problem for the carrier. Even though they themselves may not be affected, there is an increased chance that there will be reproductive consequences due to the child receiving an unbalanced chromosomal complement. The nature of these consequences depend on the particular chromosomes involved and the size of the translocated material. It is not possible to outline all of these in the fact sheet. It is, however, important for the translocation carriers and their partners to seek specialist genetic advice prior to conception. Let's see the role in the disease. Some human diseases caused by translocations are cancer. Several forms of cancer are caused by acquired translocations as opposed to those present in the conception. This has been described mainly in leukemia, acute myelogenous leukemia and chronic myelogenous leukemia. Translocations have also been described in solid malignancies such as Ewing's sarcoma. And let's see also in infertility. One of the would-be parents carries a balanced translocations where the parent is asymptomatic but conceived fetuses are not visible. Down syndrome is caused in a minority, 5% or less, of cases by a Robertsonian translocation of the chromosome 21 long arm onto a long arm of chromosome 14. Chromosomal translocation between the sex chromosomes can also result in a number of genetic conditions such as XX male syndrome, caused by a translocation of the SRY gene from the Y to the X chromosome. By chromosome denotation, the International System for Human Cytogenetic Nomenclature (ISCN) is used to denote a translocation between chromosomes. The designation T denotes A, B and P1 and Q2 is used to denote a translocation between a chromosome A and chromosome B. The information in the second set of the parenthesis, when given, gives the precise location within the chromosome for the chromosomes A and B respectively, with P indicating the short arm of the chromosome and Q indicating the long arm, and the numbers after P and Q refers to the regions, bands and subbands seen when staining the chromosome with the staining dye. The table indicates the translocations and their associated diseases with the fusion genes and their proteins. T814 with Q24, Q32. Associated disease is Burkitt's lymphoma and the fusion genes are CMYC on chromosome 8, gives the fusion protein lymphocyte proliferative ability. And in the second, IgH at immunoglobulin heavy locus on chromosome 14 induces massive transcription of fusion protein. In next case, T1114 with Q13 and Q32. The associated disease is mantle cell lymphoma and the fusion genes and proteins are cyclin D1 on the chromosome 11, give fusion protein cell proliferative ability. In the second being IgH at immunoglobulin heavy locus on chromosome 14 induces massive transcription of fusion protein. Number 3, T1418 with Q32 and Q21. The associated disease of follicular lymphoma, nearly 90% of the cases, and the fusion gene and proteins indicate IgH at immunoglobulin heavy locus on chromosome 14, inducing massive transcription of fusion protein. On the second, BCL2 on chromosome 18 gives fusion protein anti-apoptotic abilities. And in the fourth case, T10 various, Q11 various. The associated disease are papillary thyroid cancer and the fusion protein are RET proto-oncogene on the chromosome 10 and the second being PTC papillary thyroid cancer, placeholder for any of the several other genes and proteins. And in fifth case, it is T23, T23, Q13 and P25. Associated disease is follicular thyroid cancer and the fusion protein and genes associated are PAX8, paired box gene 8 on chromosome 2. In the second case, it is PPAR gamma 1, peroxisome proliferator activator receptor alpha 1 on chromosome 3. In the next case, it is T821, Q22 and Q22. Associated disease being acute myeloblastic leukemia with maturation and the gene involved are ETO on chromosome 8 and in the second case it is AML1 on chromosome 21, found in nearly 7% of the new cases of AML, carries a favorable prognosis and predicts good response to cytosine arabidinose therapy. In the next case, T922 Q34 and Q11P, Philadelphia chromosome. The associated disease is chronic myelogenous leukemia (CML), acute lymphoblastic leukemia (ALL), and the gene associated are ABL1 gene on chromosome 9. In the second case, it is BCR, breakpoint cluster region on the chromosome 22. In the next case, it is T1517 with Q2221, the associated disease being acute promyelocytic leukemia, and the genes involved is the PML protein on chromosome 15 and the second being RAR alpha on chromosome 17. Persistent laboratory detection of the PML RARA transcript is strong predictor of relapse. In the next case, T12 and 15 between P13 and P25, associated disease being acute myeloid leukemia, congenital fibrosarcoma, secretory breast carcinoma, memory analog secretory carcinoma of salivary glands. The gene associated here is TEL on chromosome 12, the second being TRKC receptor on chromosome 15. In the next case, it is T9 and 12 with P24 and P13. The associated disease is CML and ALL, and the gene involved are JAK on chromosome 9 and the second being TEL on chromosome 12. In the next case, it is T12 and 21, P12 and Q22. The associated disease is ALL, and the genes involved are TEL on chromosome 12 and AML1 on chromosome 21. Yet in the next case, it is T11 and 18, Q21 and Q21, and the associated diseases are MALT lymphoma, with the gene involved are BCL2 and the second being MLT. And the next case, T1 and 11, Q42.1 and Q14.3. The associated disease is schizophrenia and the gene involved are not mentioned. T25, T25 and P23 and Q35. Anaplastic large cell lymphoma is the associated disease and the genes involved are ALK and in the second being NPM1. T1122, Q24 and Q11.2 to 12, the associated disease being anaplastic large cell lymphoma with the genes involved ALK, NPM1. T1122, Q2411, 2 and 12, the associated disease being Ewing's sarcoma and the genes involved are FLI1 and EWS. And in T1720, 22, the associated disease are DFSP and the gene involved are collagen 1 on chromosome 17 and the second being platelet derived growth factor B on chromosome 22. And in the last case, it is T1 and 12 with Q21 and P13. The associated disease is acute myelogenous leukemia and the genes are not given here. And in the last case, T1716, Q3234 and P11 or T1116 with P11 and P12. The associated disease is low-grade fibroxymoid sarcoma with the genes involved are FUS and the second being CREB3L2 and CREB3L1. The results of having an unbalanced translocation. Autosomal reciprocal translocations, also called as translocations between the chromosomes number 1 and 22. The major reproductive risk for couples where one partner has a balanced autosomal reciprocal translocations or miscarriages. The translocation seems to be imply no risk on the body, on the baby, but a woman may have a repeated miscarriages before attaining a pregnancy. Having a child with an unbalanced translocation, the chance that carrying a balanced translocation will cause a genetic condition in the child due to receiving unbalanced chromosomal material varies from less than 1% to up to 20%, according to the specific chromosomes involved in the translocations. Translocations between an autosome and an X chromosome. Where a woman has a translocation between one of her autosomes and one of her X chromosomes, there is a probability that she will be infertile. If she is able to achieve a pregnancy, there is a high risk of the baby having problems due to the child inheriting an unbalanced chromosome complement. The degree of the severity of the problems will again depend on the specific autosome and the breakpoints involved in a man who carries a translocations between one of his autosomes and his X chromosome is likely to be infertile. This male has Down syndrome of the translocation type due to having a total of 3 copies of the chromosome 21, 2 individual copies and a third copy attached to the chromosome 14. This is according to the sources of Sales Genetic, Prince of Wales Hospital, Randwick. The risk of having a child with a problem varies according to the specific chromosomes involved in the translocations. Generally, when a mother is a carrier of the balanced Robertsonian translocations between the chromosome 14 and 21, there is a substantial risk of the child having a Down syndrome of the translocation type. Uses of translocation. Genome restructuring by translocations. The exchange of chromosome parts between non-homologous establishes new linkage relations. These new linkages are revealed if the translocated chromosomes are homozygous, and as well as we shall see even when they are heterozygous. Furthermore, translocations may drastically alter the size of the chromosome as well as the position of the centromere. Genome restructuring by translocations. The above picture indicates the phenomena. Short arrows indicate breakpoints in one homologue of each of the 2 pairs of acrocentric chromosomes. The resulting fusion of the breaks yields one short and one long metacentric chromosomes. In heterozygotes, having 2 translocated chromosomes and their normal counterparts, there are important genetic and cytological effects. Again, the pairing affinities or homologous regions dictate a characteristic configuration for chromosome synapsed in meiosis. As a result of the equality of the adjacent and alternate segregations, half of the gametes will be incapable of contributing to the next generation. This condition is known as semi-sterility. The condition of semi-sterility or half-sterility is an important diagnostic for translocation heterozygotes. Translocations are economically important. In agriculture, translocations in certain crop strains can reduce yields considerably owing to the number of unbalanced zygotes that form. On the other hand, translocations are potentially useful. It has been proposed that insect pests could be controlled by introducing translocations into their wild populations. According to the proposal, 50% of the offspring of the crosses between the insects carrying the translocations and the wild type would die, and 10 out of 16 of the progeny of the crosses between the translocations bearing insects would die. Translocations, inversions and deletions produce partial sterility by generating unbalanced meiotic products that they themselves may die or they may cause zygotes to die. Translocations are also used to isolate the human gene from neurofibromatosis. Once again, the critical chromosomal material that come from the people who not only had the disease but also carried chromosomal translocations. The translocations all had one of their breakpoints in chromosome 17 in a band close to the centromere. Hence, it appeared that this band must be the locus for the neurofibromatosis gene, which had been disrupted by the translocation breakage. Subsequent analysis showed that the chromosome 17 breakpoints were not identical and their positions help to map the regions occupied by the neurofibromatosis gene. Isolation of DNA fragments from this region eventually led to the discovery of the gene itself. Now let's select two different reciprocal translocations of the same two chromosomes. Two translocations has a breakpoint somewhere in heterochromatin and each has another breakpoint in euchromatin, non-heterochromatin material, on opposite sides of the region, and we want to duplicate or delete as shown in the above figure. It can be seen that if you have a large collection of the translocations having one heterochromatic break and a euchromatic break at many different sites, then duplications and deletions from many parts of the genome can be produced at will for a variety of experimental purposes. More generally, if one breakpoint of a translocation is near a dispensable tip, then duplication or deletion of this tip can be ignored, and the translocation can be used as a way of generating duplications or deficiencies for other translocated segments.